What is gas chromatography mass spectrometry, GC-MS? Every sample tells a story, and often that story is hidden inside tiny peaks on a chromatogram, gas chromatography mass spectrometry, or GC-MS is the tool that allows us to unlock those stories, revealing the molecular identity of even the smallest traces of chemicals. In this session, we'll explore how GC-MS separates, identifies, and confirms compounds with powerful precision, and why it remains one of the most valuable analytical tools across modern science. GC-MS is one of those instruments that quietly influences nearly every scientific field. In crime labs, it helps investigators detect accelerants, identify toxins, and analyze explosive residues. In pharmaceuticals, it ensures API purity, uncovers residual solvents, and even detects nitrosamines in drug products. Environmental scientists rely on GC-MS to monitor VOCs, pesticides, and industrial pollutants in air and water. And the beauty of GC-MS lies in one simple truth. If something can be vaporized, GC-MS can probably detect it with remarkable precision. To understand GC-MS, imagine you have a jar filled with mixed beads. First, you sort the beads by size. Then, you scan each bead to read the tiny code engraved on it. That's exactly how GC-MS works. The gas chromatography portion sorts molecules based on volatility and interaction with the column. The mass spectrometry portion reads each molecule's unique fingerprint. Together, they don't just separate compounds, they provide accurate identification and confirmation. In the GC section, everything begins with sample injection. As soon as the sample enters a hot inlet, the compounds vaporize instantly. Once vaporized, the molecules travel through a long, coiled column carried by an inert gas. Inside this column, each molecule interacts differently with the stationary phase. Fast-moving molecules have minimal interaction, while slower ones interact more strongly. This difference in interaction creates separation, and the compounds exit the column as clean, distinct peaks. GC is essentially the racetrack where molecules compete to reach the detector. Once the compounds leave the GC column, they enter the mass spectrometer, the identity decoder. First, the molecules are ionized, often by electron impact or other soft ionization methods. Next, they fragment into ions with distinct mass-to-charge ratios. Then, the mass analyzer separates and measures those ions, producing a mass spectrum. Each spectrum is like a fingerprint, unique, reproducible, and highly diagnostic. And using reference libraries such as NIST, we can match unknown spectra to known compounds with confidence. On their own, GC and MS are powerful, but when combined, they become unmatched. GC separates mixtures that contain compounds with different boiling points or polarities. MS identifies those compounds with high accuracy using fragmentation patterns and spectral databases. With GC-MS, we can separate complex mixtures, identify compounds confidently, quantify trace impurities at parts per billion levels, and confirm identity using spectral fingerprints. This is why GC-MS is widely considered the gold standard for analyzing volatile and semi-volatile compounds. GC-MS is used in some of the most impactful industries today. In forensic toxicology, it detects alcohols, drugs, poisons, accelerants, and explosive residues. In pharmaceuticals, it protects patient safety by checking API purity, identifying residual solvents, and detecting impurities. Environmental scientists use it to monitor VOCs, pesticides, and industrial contaminants. In food and beverage manufacturing, GCMS ensures flavour authenticity and screens for contamination. Even the cannabis and natural health industries rely on GCMS to quantify terpenes, cannabinoids, and solvent residues. Wherever chemical identity matters, GCMS plays a vital role. Behind every great GCMS result is thoughtful method development. Sample preparation is everything, because even the best instrument can't fix a contaminated or poorly prepared sample. Choosing the right column is crucial. Length, inner diameter, film thickness, and stationary phase all influence separation. Inlet conditions, including temperature, split ratio, and liner type, directly affect peak shape and reproducibility. And never forget ion source maintenance. A dirty source means noise, drifting baselines, and poor sensitivity. These small decisions are what transform an average chromatogram into a scientifically valuable one. As powerful as GCMS is, it does have limitations. Not all compounds are volatile or thermally stable enough to survive GC conditions. Some molecules require derivatization, which adds extra steps and complexity. Columns degrade over time, which means recalibration and maintenance are essential. Despite these limitations, when GCMS is used correctly, it remains unmatched for analyzing volatile and semi-volatile compounds. GCMS is evolving rapidly. We're seeing faster analyses using high-speed columns, higher-resolution mass analyzers that reveal new spectral detail, and robotics that automate sample preparation. Portable GCMS units are now used for on-site forensic analysis, and with AI-assisted spectral matching, data interpretation is becoming quicker and more accurate. Even as the technology evolves, the core strength of GCMS remains unchanged. Its ability to reveal what's hidden within complex mixtures with striking clarity. GCMS doesn't just give data, it gives answers.